Okay, so welcome back. We're going to start a new unit today, and I just want to put it in the context of what we've learned before in the previous units. So remember, in our first unit we had an introduction to the members of the microbial world, as well as an introduction to infectious diseases, and kind of set the stage for learning about differences between pathogenic and non-pathogenic microorganisms and their role in human disease. And in the second unit, we learned about the structure and function of microorganisms, in particular, differences between prokaryotic and eukaryotic microorganisms, and the basic features of the structure of bacteria in particular, and some of these key features that are associated with virulence. Then in the third unit, we learned about the growth of bacteria and control of bacteria, as well as not just the growth, but also the metabolism that is a part of that growth of bacteria. And differences in the kind of very large diversity that we see with bacteria, being that they're pretty much ubiquitous in our environment here on Earth, so they have very diverse capabilities depending on which species you're talking about. But the importance of control and understanding control in the context of those microorganisms that are pathogenic and that we want to limit their presence in order to prevent disease. So now in understanding control and also in understanding treatment of infection, not just prevention, we're going to move on to a discussion of antibiotics. But before we do that, we really need to understand more about the genetic makeup of microorganisms, and I'm going to focus on bacteria, not just their genetics, but also gene expression and ways that these genes can be controlled or expressed selectively in order to adapt to the environment. And this ties in also not just with susceptibility to antibiotics, but with resistance to antibiotics. And I'm going to touch a little bit upon biotechnology, but not too much, more just to understand some of the lab techniques that are available for identifying bacteria and classifying them. But if you want to learn more about biotechnology, there are some great biotechnology courses offered here at Bunker Hill. Okay, so Chapter 8 reviews in more detail and gives more detail about the genetic makeup of microbes, in particular bacteria, but also key differences between prokaryotic microbes like bacteria and eukaryotic microbes such as fungi. And we'll go over the structure of the genome and the components of the genome, as well as some ways that the genome can be modified or regulated in order to adapt to the environment. Here again, as usual, is a list of terms that you should be familiar with after reading the chapter and listening to this lecture. So I have to preface this with a note that this should really sound familiar to you. It should be a review from a general biology course or the anatomy and physiology course that's a prerequisite for this course. So I'm going to move pretty quickly through it. So DNA, the structure, as we already mentioned earlier in the course, it's a double helix. It runs in two different directions. We call it antiparallel, but and it's held together. These two strands are held together by base pairing, which are hydrogen bonds formed between the bases of adenine and thymine, or cytosine and guanine. And this structure is where, and the sequence of these nucleotide bases is really where we find the information, genetic information stored. So all of those traits that an organism has are determined by the genetic sequence within its DNA. So the genome is all of the DNA in an organism, whether it's a microorganism or a macroorganism, but in the case of microorganisms, for example, all of the DNA in a bacterium that's required to make a complete new bacterium. Okay? And the genome includes both those genes that are needed to make a whole nother organism, as well as additional genes that can be beneficial for survival in certain environments or under certain conditions. All right, so as I mentioned, the DNA is where genetic information is stored. That's all of the sum of all of the traits that an organism has is determined by the genes that it carries. And those genes need to be expressed as their protein products or RNA products in order for the organism to be alive. So the genes carry a sequence of bases: adenine, thymine, cytosine, and guanine. And as I already mentioned, the A or the adenine pairs by making hydrogen bonds with thymine, and guanine makes actually 3 hydrogen bonds as opposed to 2 hydrogen bonds between the A and the T. The guanine and cytosine, or the G and C, form 3 hydrogen bonds, and that's really what holds the double helix of the DNA together, but also allows it to come apart, for example, when replication or gene expression needs to occur. So usually, let me just go back for a moment. Usually bacterial genomes have a chromosome, a single chromosome that consists of this double-stranded helical DNA that carries all of the essential genes that make up that particular species of bacteria. And the gene is just a unit or a part of that DNA that can be expressed in the form of an RNA intermediate, and that RNA intermediate, the messenger RNA, then by other mechanisms in the cell is converted to a functional form, i.e. a protein, a specific protein. The process of the gene, the information in a gene, being converted to a useful product, i.e. a protein, is called gene expression. So let's just take a moment to look at the bacterial genome in a little more detail and go over some examples. So E. coli, Escherichia coli, is very easy to grow in the lab, and so it's been a focus of a lot of research on understanding the genome of bacteria. And here's a map. The entire genome of E. coli has been sequenced and it's been mapped out, and so the colored blocks in this circle, the purple circle, which represents the single circular form of the chromosome, so these colored blocks represent genes within, or clusters of genes within that genome. And you can see that this genome, actually you can't see the final number, but it is 4.6 million base pairs. So base pairs are those matching A, T's, or G's and C's that make up the entire genome. And this is just an example. It's not that every microorganism has a genome that size. In fact, the genome size varies from very small, around 1 million base pairs, to very large, around 9 million base pairs. And when you have a larger genome, for example, Pseudomonas aeruginosa can have a genome as large as 7 million base pairs, and that additional DNA means that it has additional genes within that genome that allow it to be more versatile. In the case of Pseudomonas, it's able to grow in a variety of environments because it is not solely dependent on glucose as a source of food, but instead can also use amino acids and break down about 80 different substrates because of these additional genes that encode for enzymes that can be secreted and then break down the products in these substrates in the environment of the Pseudomonas bacterium and allow it to be used in the metabolism of those bacteria. On the other hand, your book has a table giving examples of a variety of different species, but something like Borrelia burgdorferi, which we've mentioned before, the causative agent, the bacteria that causes Lyme disease. I mean, these bacterium have a genome size of just over 1 million base pairs, so quite small. And it's unlike E. coli and many other types of bacteria, it's not a circular genome, it's actually 22 segments, so linear fragments of DNA. And it's not as well understood how that genome is replicated, but it is understood that these genes within these segments help the bacterium to switch from living in a tick to living in a deer or living in a human host. So, as I mentioned, the organization is a single circular chromosome. Within that chromosome, there are clusters of genes. So you can see by the same color, that's a cluster of genes that we call operons that are associated with the same function. So here in yellow, membrane synthesis. There are a variety of genes kind of scattered throughout the genome for DNA replication and repair. But then there's a cluster for lipid metabolism and a cluster in the red box for carbohydrate metabolism. And the clustering of these genes into operons, we'll see later in the lecture, is really key for the ability of the bacteria to regulate or turn on and off certain features of their metabolism. And we'll talk about regulation of gene expression, but many genes are constitutive, i.e. they're essential and have to be expressed all of the time in order for the bacterium to survive. But there are other important genes that are repressed until they are needed or induced when they're needed. So the genes within the genomes are organized, and within these operon regions there are other parts of the DNA that are not encoding a product but are essential to the proper function of the organism. So regulation, when these genes are turned on and off, or how they're repressed, or how they're induced, is determined by the upstream region, or the region on the 5' side of the genes. So these include important sites like an operator site, where a regulatory product known as a repressor may prevent the enzyme required to make the messenger RNA, the RNA polymerase enzyme, from proceeding to the actual gene or set of genes within the operon. And the RNA polymerase enzyme binds and recognizes the promoter with the help of general transcription factors. And you can see here upstream from the operon is actually a single gene under the control of a single promoter, which is really the only kind of genes that we find in eukaryotic cells, such as our own, such as microorganisms that are eukaryotic, such as algae or fungi or helminths. So these operons, these organizations within the bacterial chromosomes, are really unique to prokaryotes. And what they enable the bacteria to do is make a set of genes that's, say, for example, necessary for switching from metabolizing glucose to metabolizing a different carbohydrate that may be present in their environment, so they can survive longer. And that may involve several genes in order to, for example, transport that, break down that carbohydrate into its simple form so that it can be glucose or transported into the cell, etc. And as I mentioned, bacterial genomes differ from eukaryotic genomes because not just because there's a single promoter associated with a single gene, but there are also other regions, other factors that are different. So in bacteria, the genome is more simple. It just in that gene has all the information that it needs to make it into a protein. So it's all coding information in that sequence of nucleotides within a gene. But that's not the case for eukaryotic genes. Eukaryotic genes have both exons, which are the coding part, and introns, or intervening segments of sequences of these nucleic acid bases that are not part of the information needed to make the messenger RNA, and so there are extra steps that need to occur in eukaryotes before that messenger RNA is mature and ready to be translated into a functional form by the ribosome in the process of translation to make a protein, a functional protein. So bacteria are simpler. Remember, their structure is also simpler. They don't have a separate compartment where you find the DNA. Instead, it's in a region kind of concentrated within the bacterium, but it's readily accessible to enzymes that are necessary for expression, like RNA polymerases, as well as enzymes that are necessary for making proteins, like ribosomes. So we'll see in a minute that this can enable the bacteria to express genes in a much faster manner than in eukaryotic microorganisms. But first I want to touch upon another form of DNA that you find in bacteria that we already learned a little about in Chapter 4, but I want to talk about it again and bring it to your attention again. And that's plasmid DNA, or what we call extrachromosomal DNA. So the whole genome includes the chromosomal DNA as well as any extra DNA in the form of plasmids. And they're called extrachromosomal because they're actually, if the bacteria loses that plasmid DNA and only has the chromosomal DNA present in a daughter cell, it can still survive and live and replicate and metabolize. So it's kind of all bonus information that's carried in the genes of plasmids, but it's really key for traits such as resistance to antibiotics. So the plasmid DNA is much smaller. It's anywhere from 0.1 to 1% of the size of the genome. And so it's able to actually enter or be passed from one bacterium to another um, without um, changing the species of a cell, and it's small enough that it can be successfully transferred in its entirety through these interactions. And we'll learn more about this type of transfer of plasmids from bacterium to bacterium in Chapter 9. But within the genes that are carried on this small fragment of DNA, um, they can be useful. They can be virulence genes. They can confer added survival advantages. As I mentioned, antibiotic resistance can also be resistance to disinfectants. Um, so even though it's a small number of genes, they can be really important for um, the survival or the advantage of a pathogenic microbe organism or pathogenic bacterium. Um, and in addition, within the bacterium, these plasmids are independent of the chromosomes. So they will be replicated as the chromosome is replicating, but they can also replicate themselves. They have their own origin of replication, so they can replicate themselves even when the chromosomal DNA is not being replicated. So you can get many copies of the same plasmid within a single bacterium as a result, and that can change the amount of gene expression from very low level to a high level of the products of these genes being made. And one example of a plasmid that carries drug resistance is the plasmid present in methicillin-resistant Staphylococcus aureus, which you may have heard of as referred to as MRSA. Not only is just methicillin resistance encoded by the genes carried by this plasmid, but also other antibiotic drugs within the penicillin family, as well as um, a few resistance to a few disinfectants. So it's really more than just methicillin resistance that a bacterium is gaining when it gains the resistance or R plasmid to form this new strain within the species of Staphylococcus aureus. And I'll go over the difference between strain and species a little bit later in my lecture. So a big part of understanding, of my review of this understanding of what DNA is in microorganisms like bacteria is to understand how these bacteria are able to adapt to their environment. Why are they so hardy? There's lots of changes in the environment that are going on throughout their lifespan. So how are they able to survive, and how are they able to, for example, change over time to become resistant to antibiotic drugs? Well, one way that the species is maintained is by reproduction, right? So we already learned that bacteria reproduce by binary fission. It's kind of like making a clone of itself over time. And so that entire chromosomal DNA, in fact, the entire genome, if it's including both chromosomal and plasmid DNA, is inherited by the offspring of the parent cell. But in many cases, and in particular in E. coli, depending on the environment, there may be mutations, and the environment may enhance the number of mutations. So there's an inherent error rate in DNA replication that's about one error in every 10 million base pairs that are replicated. So very, very low, very rare event. Because remember that the bacterial a chromosome in the case of E. coli is about 4.6 million base pairs. So it's got a pretty good rate of making of maintaining the integrity of the DNA in this process of replication. But that can be modified depending on the conditions. If there's a mutagen present in its environment, then that rate can increase and be more common. So if we just take an example of a colony of bacteria, remember E. coli have just under 5 million base pairs in their genome. It's about 4.6. So, but remember that in a colony you have millions of bacteria that become visible to the naked eye because there's so many bacteria growing in that community. And so about half the bacteria in that community should contain a single mutation, a single difference. So that doesn't mean that they're different species, they're just a variation on the species that we call strain differences. And it may not even be a strain difference because it may not result in a change in the traits of that bacterium. So before I go on to talk about these changes due to mutations, I just wanted to review what DNA replication is like, especially in bacteria. So the process of DNA replication occurs in bacteria during binary fission when the bacteria are dividing. And at first, a complete copy is of the DNA of the genome is made. That usually in the form of a single chromosome, a complete copy is made. As at the same time the cell is enlarging and more cell wall is being made, and then the process of septation occurs after the DNA is fully replicated to separate the two cells. So during DNA replication, there are several things that have to happen in order to ensure that this is really a copy and the integrity of that copy is good, like it's an accurate copy. Okay. One thing, and many of these processes during replication help support that. So one thing that happens during DNA replication is that it's semi-conservative, and what that means is that one strand is used as a template while a complementary strand is being synthesized into this new polymer of DNA. And in bacteria, because the genome is usually in the form of a single circular form, that circular form of the genome means that the DNA replication can happen in two directions at the same time. And so in that sense, there are two templates that are being used to make two new strands, and in the end you end up with two complete double-stranded DNA forms of the genome. So there's different ways that the polymerization occurs depending on which direction the enzymes are going. There's a leading strand that just continuously polymerizes base after base that's complementary to that template, and then there's the lagging strand that has to go in short spurts, and so it polymerizes short fragments of the DNA, and then those fragments, called Okazaki fragments, are then ligated together by a separate enzyme to form a complete second complementary strand of DNA in the process. So how do these enzymes know where to begin? They have a certain place within the genome that marks the start site called the origin of replication. And remember that plasmid DNA can be replicated independently from the chromosome or chromosomal DNA because it has its own origin of replication that can be recognized by the enzymes that carry out DNA replication, the DNA polymerases. And that origin of replication marks the start, and the terminus marks the stop site of replication. So the DNA polymerases are the enzymes that physically form that covalent link between each base and the growing polymer of the DNA. And those bases have to be complementary, able to form these non-covalent interactions with the template strand in order to be incorporated into the growing new strand of DNA. In eukaryotes, there are more than one type of DNA polymerase, and they have different functions. DNA polymerase 1 being the proofreading form of the DNA of the enzyme, but in eukaryotes we have a simpler system. There are, of course, other accessory enzymes that are really required for this whole system to work. And finally, I'll mention a couple of modifications that occur after the genome is copied. So, as I mentioned, a special case for bacteria is that they can have a bidirectional replication in which a new strand of DNA is made here in this diagram depicted as the purple ribbon from using the original strand, the parental strand here depicted in a gray ribbon of the DNA. And this diagram also shows that the process is starting at the origin of replication. And this would require two separate DNA polymerase enzymes to be moving in separate directions along this DNA. But they're always polymerizing in the 5' to 3' direction, whether they're going off to the right or to the left. And what's not shown here are those enzymes, in fact. So the enzymes, they start at the origin and they end at the terminus. This bubble that forms is a separation. So the hydrogen bonds that are holding that DNA double helix together are broken in the presence of the DNA polymerase. And that enables each strand to be functioning as a template. So the enzymes move in opposite directions as the replication fork opens. And the leading strand is a continuous strand. So that's replicated in the 5' to 3' direction. And the lagging strand is also replicated in the same direction, but in kind of fits and spurts in that it's being made in these short fragments of nucleotides called Okazaki fragments. And those are kind of pieced together. So in that way, even though the terminus is not exactly at the origin, you're getting a complete copy. So the lagging strand is really going in the opposite direction of the leading strand, but because it's still polymerizing in the same direction, it has to kind of start polymerizing and then it stops and then it goes back again and polymerizes another fragment. So as the fork opens, it polymerizes these short fragments. And that happens in two directions, where you're having the leading and the lagging strand being polymerized, so that in the end you've got 4 strands of DNA, 2 complete copies of the genome by the time the enzymes get to the terminus. In the end, you have the parent strand here in black and the newly synthesized strand in purple. And there are other enzymes like ligases that fill in all of these gaps that you see between the Okazaki fragments to make a complete copy. And then the way that the cell can tell between the leading and lagging strands, so if there's any mistake made along the way in this the way that the cell can tell between the parent and the newly synthesized strand is important, because any mistakes that are made, it has to be able to tell which one was the original template strand and which one being the correct sequence, and then which one is the strand with the mistake. And the way it can tell is that DNA is methylated, but the newly formed strand is not yet methylated. So after the completion of the synthesis of the newly formed strand, then methylation occurs, and the separation of the 2 complete genomes occurs, as well as a modification. So there has to be an unwinding. There's some tension that builds up in the twisting or the helical shape of the DNA, and that tension has to be released in order for the DNA to be able to be expressed, for the genes to be able to be expressed. And that's that those negative supercoils, or that kind of unwinding, is accomplished by a different set of enzymes, gyrases, and there are 2 major gyrases, A and B, in bacteria, that carry out this kind of, they cleave both strands of the DNA, which requires energy in the form of ATP, and then they catalyze the unwinding, and then religate the 2 strands. And why I'm mentioning this in particular is that these enzymes are target of a class of antibiotics called quinolones, which are sometimes used to treat infections, bacterial infections. And it's a selective target because these enzymes are present in bacteria but not present in our own eukaryotic cells. I'm giving you this information because it's relevant to the mutations, the mutation rate, and during this process of replication, there is a proofreading function that enzymes carry out in order to ensure that there's only rare cases, there's a mistake made in copying the genome. Because if there are too many changes, it would result in malfunction of the products of the genes, and so the microorganism wouldn't be able to survive. But also it means that because these enzymes are not perfect in every case, although they're rare, but it means that changes can result in these rare cases that could lead to a different strain within the same species. So during DNA replication, there's about 1 error in every 1,000, but those errors are often corrected by these DNA repair enzymes that are able to detect mismatched bases, so they're not properly hydrogen bonding, they're not lined up properly within the DNA double helix to be able to hydrogen bond, and so they're recognized and they're repaired. And DNA polymerase I is one of those enzymes that detects those mistakes. And with the help of these repair enzymes, they are corrected. So, as I mentioned, I'm introducing this information and reviewing this information about the genome of bacteria in order to understand how they change within a changing environment, how they adapt, how they respond to their environment. And so they have basically three different ways that they're able to change and maybe survive when their environment changes. So in many cases, the environmental changes will mean that many bacteria die, but some of them may have acquired a mutation that allows them to survive these changes in the environment. Some of them may be able to switch a new set of genes on in order to, for example, use a new carbohydrate as a substrate for growth when glucose runs out. And that's regulation of gene expression. They're turning on and off of genes. And then finally, gene transfer, or the sharing of genetic information between bacteria. And there are a variety of ways that that can happen. I'm not going to go over that until we talk about it in Chapter 9. So let's go back to mutation. I mentioned the rate is very, very rare, 1 in 10 million base pairs. But it's never perfect. So that enzyme, DNA polymerase, that's responsible for making a complete copy of the bacterial genome, just as it is in our own human cells, does occasionally make mistakes, and those mistakes, while many of them can be repaired by mechanisms of repair, some of them aren't repaired, and so they're passed on to the daughter cells. And by definition, a mutation is anything that is inherited by those daughter cells. So any change or alteration in the DNA sequence, regardless of whether it's good, bad, or doesn't really change the function or traits of an organism. But sometimes these changes are advantageous and they become the majority of the population. But regardless of whether that happens, they're always inherited by the daughter cells during the process of binary fission. So that's what we call vertical gene transfer and reproduction. And I'll come to this in just a minute again. And then there are two basic requirements that in order for a mutation to be inherited by a daughter cell. One is the base sequence, so that sequence of nucleotide bases, the A's, the G's, the T's, and the C's, it changes, and the cell mechanisms of repair fail to correct it. Okay? So here's a variety of possibilities that can occur as a result of a single base change that's not corrected. Okay, here on the top left you have the missense mutation, where because the code of the DNA is 3 bases determine a single amino acid in the protein product, if you have a change in that final third position, you can have a change in the actual amino acid that goes in that place, in that position within the protein, and it actually makes a different protein because the amino acids are determining the shape of that protein, and the shape is what determines the function. If the shape changes, it may be that the protein no longer functions. If you have a nonsense mutation, that's often at the second position of the triplet code, in which a base is changed, right? That results again in a change in the amino acid at that position in the protein. But in this case, it's not a different amino acid, it's a stop codon. That would mean that all the rest of the amino acids that are supposed to go into that protein are no longer inserted in by the ribosome, because the ribosome is reading this these three or this triplicate code of bases as a stop signal that it shouldn't go ahead and add any more amino acids to the growing chain of the peptide, of the protein. That can have very dramatic effects on the function of a protein because you get a much shorter protein. A whole bunch of the protein is missing. Most likely that protein is not functioning. And then sometimes there's not a change in the base, but actually a removal of the base. And that leads to a shift in the reading frame. So the triplicate code, all of a sudden, when one triplicate, instead of having 3 amino acids in its place, has only 2, it starts reading the third amino acid that should be there from the next part of the DNA sequence. And so everything shifts and you get a whole new set of amino acids that are encoded from that point on. Here the lysine, you can see instead of serine, arginine, isoleucine, you're getting a lysine followed by a tyrosine, proline, and glutamine. And that, again, those different amino acids means the protein will have a different a shape different than it was intended by the gene. And so that mistake can lead to a very different functioning protein or a non-functional protein, which can affect the viability of the organism dramatically. There can also be, instead of a deletion leading to a shift in the frame, the reading frame, there can be an insertion, so an extra base inserted in during replication can again lead to a similar problem of you getting a completely different protein after that point in the amino acid sequence. And then finally, a kind of flip-flop of the inversion mutation can result again in a completely different set of, or at least 2 completely different amino acids being replaced by in the triplicate code of that region of the protein, and can result in a different protein or non-functional protein. So really these mutations are changing the protein. Sometimes if you have a change in the final or first, second, or third position of this triplicate code, you can get a nonsense mutation. You can get a mutation that results in no change in the function of the protein, i.e., because there are more than one set of nucleotide bases, so TTT, perhaps TTC, also encodes for the amino acid phenylalanine. So in that case, you wouldn't see a different protein. You would see the same protein. And that's what we call, not a nonsense, but a silent mutation. And silent mutations don't change the function of the protein. All of these other examples do change the protein, the shape, the length, the function of the protein, but silent mutations do not change anything about the protein. It's just the same as it was, even though there's been a change in the nucleotide base sequence. All right, so sometimes mutations, so usually when mutations like we just talked about occur, there are mechanisms, or proofreading mechanisms that DNA polymerase has to go back and make sure that the base that it inserted in the new growing strand of DNA matches that template strand, that original strand of DNA. And so it goes back and there's a set of enzymes and ligases that will correct that, basically cut out and correct that base that's mistakenly polymerized in the chain of DNA. But sometimes the mistake fails to be repaired, and so it becomes a mutation, becomes inherited by subsequent daughter cells. There are agents that can be present in the environment of the bacteria that can induce induce changes in the DNA. So physical agents that can be mutagens include X-rays, UV radiation, that make it uh, that make the DNA polymerase um, result in an incorrect insertion of the incorrect base, or unable to read the base properly. So pyrimidine dimers, like a thymine-thymine dimer, where the two thymines, instead of being properly hydrogen bonded to the other strand, they're covalently bonded to each other when they're together in a sequence on one strand of the DNA. Can really wreak havoc with the ability of DNA polymerase to correctly replicate that template strand. There are also chemical agents that might be present in the environment of the bacteria that can also be mutagens. And if these physical and chemical agents, so let me just go back for a moment. These chemical agents include hydrogen peroxide, so very highly reactive molecules, superoxide, any kind of oxygen radical like superoxide or hydrogen peroxide are going to be very reactive and be readily giving up electrons, and so they tend to not just disrupt DNA structure, but also disrupt protein function. And there's other kinds of dyes that are mutagens, and many of these are also carcinogens. They're not just changing bacterial DNA, but they can also change our own human DNA. So you have to be careful when you are working with these agents. So we all know, sunburn by UV radiation from the sun can also cause similar mutations in our own cells that are a prerequisite for cancer, in the case of skin cancer. So, but these physical and chemical agents that are mutagens tend to speed up that kind of inherent, very rare mutation rate to the point where it can be not only causing changes to the genes within a microorganism, but killing that microorganism because too many of its genes have been damaged and the DNA polymerase is unable to repair so many mistakes. So it's actually, many of these things are means, as we learned about in chapter 7, when we learned about control, chapter 13 rather, when we learned about control of microorganisms. These physical and chemical agents may sound familiar from those chapters because they are a means of controlling microorganisms by inducing mutations which will result in the malfunction of their proteins, or the inability of their proteins to function properly, and without protein the microorganism such as bacteria cannot survive. So let's just look at the consequence though of a mutation that is inheritable. That's not many mutations, but a few rare ones that are acquired by a single member of bacterial population, but it doesn't acquire enough to actually kill the bacterium, so it survives and it's able to metabolize and replicate. And in certain environments, for example, in the presence of antibiotics, it may give that bacterium a growth advantage over the other bacteria in that population. So here you can see in this test tube, in this liquid culture, the bacterial cells are represented by these colored dots, and most of them are susceptible to the antibiotic streptomycin. But there's one that's acquired a mutation that made it resistant to streptomycin, i.e. it's changed something about the target of streptomycin which targets the ribosome. So something about that structure of the ribosome is different, so the drug can no longer bind and have its inhibitory effect. And so when the environment changes and the bacteria are trying to grow in the presence of streptomycin, all of the population is killed except for that one streptomycin resistant cell. Whereas if you just plated it in an environment without any drug, all of them would grow, and it would be a very tiny minority of the population that would have that drug resistance. But now, if you culture that in the presence, if you culture that colony that grew up in the presence of streptomycin, and you grew it again in this liquid culture, what color would all of these cells be? Well, they would only be red. They would only be streptomycin resistant cells. And each cycle of replication, each time binary fission occurred, it would again be a streptomycin resistant cell. So you can kind of imagine this is the problem that occurs in the human body when a course of antibiotics is not taken to its completion. So first you have some environmental stress that wipes out most of the bacteria, but if there's some that can survive in the presence of that antibiotic, then and the antibiotic is not continued to be taken, so it may be that they have weak resistance, and if the antibiotic had been continued to be taken for the full course, that they would die. But instead they persist, and then they become the majority of the bacteria, and the infection is not cured. They're still causing the damage that was originally causing the infection, and the reason that that patient probably went to seek help from their physician or treatment. All right, so that's mutations, and I focused more on what happens at the gene level in that case. But really we have to understand, I implied what happens to that information that's changed at the genetic level when it's expressed. So I just want to review what gene expression really is, and that's that process of information that's stored in our DNA, that genetic information that's stored in our DNA, the processing of it into a functional form. And that, by the central dogma of molecular biology, is always goes from the DNA, the gene, to the messenger RNA, that intermediate, to a protein. And this is true of any living organism, whether it's a fungi or our own human cells or a bacterium. It doesn't matter if it's prokaryote or eukaryote, this is the flow of information. And the enzymes that carry out these two key steps of transcription from DNA to mRNA and translation from mRNA to protein are RNA polymerase, which takes care of transcription, and then the ribosome, this complex of RNA and protein that takes care of translation. So you should know those two main steps and the flow of information. But in bacteria, the flow of information can be through this expression of genes, or through replication, as we already mentioned. But it can also be through recombination. So not while a bacterium is not even replicating, there can be some changes to the genome that occur through horizontal gene transfer. And that's what Chapter 9 focuses on. I'm not going to talk about it today. But let's just look at within the context of a single bacterium, not what's inherited through replication, which we call vertical gene transfer, that process of binary fission. We already touched upon, so I'm not going to go over that. Instead, I'm going to go over how expression can be manipulated by a bacterium depending on the environment, how it can adapt to that. Okay, so there are environmental sensors that will activate transcription of certain sets of genes in these organized in these operons within the genome of bacteria. And there's a kind of feedback mechanism that can occur in some cases in which a product of that set of genes, if it builds up in the cell enough, can can activate transcription by binding to the promoter and recruiting RNA polymerase to that site for that key first step in gene expression, that step of transcription. So transcription requires the RNA polymerase enzyme to put together the bases in the RNA by reading the base sequence in the DNA. And the RNA polymerase is present in the cell, and it will bind, it will touch down and kind of bounce along the DNA sequence that's exposed until it finds a really stable binding site. And that stable binding site is, it's helped to find that stable binding site by a transcription factor called sigma factor. Here it's in this cartoon it's colored red, whereas the RNA polymerase is this pale blue. So the sigma factor is what interacts with a specific sequence of bases that are upstream, so that's -10 from the gene start site, and -35. And it kind of latches on to these conserved sequences of the Tata box, which is included in that. And then it's released so that RNA polymerase can do its function of opening up the double-stranded DNA at that point and bringing in now the RNA or ribonucleotides to make that messenger RNA. So remember, ribonucleotides are a little bit different than the bases found in the DNA because there's no thymine. Instead of thymine, there's uracil that base pairs with the adenine. Eukaryotic gene expression is a little different. I'm not going to go over it. It doesn't use the sigma factor. It uses a whole set of proteins called general transcription factors. Okay, then in the second set, here on the right, you can see in this transmission electron micrograph, there are lots of black dots here, and each black dot is a ribosome. And what you can't see too well is a very pale gray strand that is in between each of these black dots. That's the DNA. And so what happens actually is sorry, it's not the DNA, it's the messenger RNA. So this is a single strand of messenger RNA that's coming off, that has many ribosomes stuck to it that are carrying out that second step in gene expression of translation. And kind of in the medium gray color is the actual protein product that's coming off of each of these ribosomes as they carry out translation. So the ribosome has some help finding the start site of within the message. There's a little bit of the message on either end, the 5' end and the 3' end, that are not translated. They're called untranslated regions. But within, just after that part, there's a conserved sequence, an ATG, where the ribosome will bind and begin its process of translation. And more than one ribosome can be attached to a single message RNA messenger RNA at the same time. And in fact, these ribosomes, as soon as the messenger RNA is made by the RNA polymerase, even before it's finished. So here you can see in the cartoon the DNA, the double-stranded DNA, and the RNA polymerase enzyme in the pale blue is doing its job of transcribing the gene into the message, which here is here in blue, bright blue. And even before that RNA polymerase has finished making that messenger RNA, there are ribosomes attaching at that ribosome binding site and beginning to make proteins. And that's something special that happens only in bacteria, because in eukaryotic microorganisms like fungi or algae or protozoa, um, there's a physical separation of the nuclear envelope that the messenger RNA has to get across before it can reach that pool of ribosomes in the cell. So there are two actually separate, step 1 and step 2 are actually two separate events in eukaryotes, whereas they're happening simultaneously. And so you get very quick process of gene to protein in bacteria, unlike eukaryotes, which takes a little bit longer because of the processing and movement necessary. Okay, so I'm not going to expect you to memorize these specific amino acids, but just to know that there's a specific start site where the ribosome recognizes and binds to the messenger RNA, and there's a specific stop site, and these are 3 codons or triplicate codes that determine which amino acid goes next in the growing polymer of the protein. In the case of these operons, the ribosome will make actually all of those genes simultaneously, and then later they'll be cleaved apart into their functional segments. And I already mentioned that this process, the 2 steps of translation and transcription and translation, are happening at the same time in the bacterium, because there's no separate compartment. The nucleoid region is accessible by the ribosomes within the cytoplasm. All right, I'm not going to go over in too much detail the phases of protein synthesis, but I do want you to understand that these phases, which break down into basically 3 steps, where there's like the first starting or initiation, then the process of making the entire polymer of the protein, so the elongation, adding each amino acid one after the next, and then termination determined by where the stop codon is in that messenger RNA. Those steps are prevented by different antibiotic drugs, which bind different components or parts of the ribosome. So here's a table from your book that shows those different targets. And remember that these antibiotics are specific for prokaryotic ribosomes. They won't affect the eukaryotic ribosome because it has a different structure. So they're going to be selectively toxic to bacteria that are causing an infection, but not be harmful to our own cells, which have the eukaryotic ribosome structure. Okay, so just quickly going down, things like aminoglycosides, like streptomycin is the one example, will bind the 30S subunit of the bacterial ribosome and block it, and interferes with that step of the elongation that requires an aminoacyl tRNA to come in. Then tetracyclines also similarly block the 30S subunit, but then they're their binding site is a little bit different, even though they're blocking the same step of the amino-acyl tRNA coming in. Macrolides, such as erythromycin, bind the larger 50S subunit of the bacterial ribosome. There are a whole bunch of other antibiotics that also bind the 50S subunit at different sites within the 50S subunit, so they have kind of different effects. Some of them are specific for all types of bacteria, like erythromycin or the macrolides, whereas others, such as linezolid, is only effective against gram-positive bacteria. So, how does a bacterium know which proteins to make and when they need to make them? This is another way that bacteria can adapt to their environment, to changes in their environment. They can turn on and off genes through the regulation of gene expression. So there's going to be a certain core set of genes that need to be on all the time. That's what we call the constitutive genes, or constitutive gene expression. Those are things that are always needed. So you always need RNA polymerase, because you're always going to need RNA polymerase to make messenger RNA so that more protein can be made from that messenger RNA in the cell. But those types of genes are not involved in the regulation of gene expression. They're just always on. But there are other genes that can be either repressed or induced depending on the conditions. And there are different levels of gene regulation. You can change the DNA sequence. In order to activate or disable a particular gene, and that's really most commonly what we're talking about is gene regulation at the level of the DNA. But they can also, as I mentioned, control the ability of RNA polymerase to carry out that first step of transcription. So maybe that there are repressors or activators that influence the ability of RNA polymerase to recognize that promoter site and bind there, or to move beyond that promoter site to the actual gene. And then there are later points where that can be controlled in the process of gene expression. Remember, first comes transcription and then comes translation. So translation can also be controlled. So initiation sequences may be bound and blocked by recognition from the ribosome. So if these repressor proteins are bound to the initiation sequence of the messenger RNA, then there's not going to be any protein made. And then after the protein is made by the ribosome, there can also be control. So before I go on to talk about these changes due to mutations, I just wanted to review what DNA replication is like, especially in bacteria. So the process of DNA replication occurs in bacteria during binary fission, when the bacteria are dividing. And at first, a complete copy is of the DNA of the genome is made, that usually in the form of a single chromosome. A complete copy is made. As at the same time the cell is enlarging and more cell wall is being made. And then the process of septation occurs after the DNA is fully replicated to separate the two cells. So during DNA replication, there are several things that have to happen in order to ensure that this is really a copy and the integrity of that copy is good, like it's an accurate copy. Okay. One thing, and many of these processes during replication help support that. So one thing that happens during DNA replication is that it's semi-conservative, and what that means is that one strand is used as a template while a complementary strand is being synthesized into this new polymer of DNA. And in bacteria, because the genome is usually in the form of a single circular form, that circular form of the genome means that the DNA replication can happen in two directions at the same time. And so in that sense, there are two templates that are being used to make two new strands, and in the end you end up with two complete double-stranded DNA forms of the genome. So there's different ways that the polymerization occurs depending on which direction the enzymes are going. There's a leading strand that just continuously polymerizes base after base. That's complementary to that template. And then there's the lagging strand that has to go in short spurts. And so it polymerizes short fragments of the DNA, and then those fragments, called Okazaki fragments, are then ligated together by a separate enzyme to form a complete second complementary strand of DNA in the process. So how do these enzymes know where to begin? They have a certain place within the genome that marks the start site, called the origin of replication. And remember that plasmid DNA can be replicated independently from the chromosome or chromosomal DNA, because it has its own origin of replication that can be recognized by the enzymes that carry out DNA replication, the DNA polymerases. And that origin of replication marks the start, and the terminus marks the stop site of replication. So the DNA polymerases are the enzymes that physically form that covalent link between each base and the growing polymer of the DNA. And those bases have to be complementary, able to form these non-covalent interactions with the template strand in order to be incorporated into the growing new strand of DNA. In eukaryotes, there are more than one type of DNA polymerase, and they have different functions. DNA polymerase 1 being the proofreading form of the DNA of the enzyme, but in eukaryotes we have a simpler system. There are, of course, other accessory enzymes that are really required for this whole system to work. And finally, I'll mention a couple of modifications that occur after the genome is copied. So, as I mentioned, a special case for bacteria is that they can have a bidirectional replication in which a new strand of DNA is made here in this diagram depicted as the purple ribbon from using the original strand, the parental strand here depicted in a gray ribbon of the DNA. And this diagram also shows that the process is starting at the origin of replication. And this would require two separate DNA polymerase enzymes to be moving in separate directions along this DNA. But they're always polymerizing in the 5' to 3' direction, whether they're going off to the right or to the left. And what's not shown here are those enzymes, in fact. So the enzymes, they start at the origin and they end at the terminus. And really this bubble that forms is a separation. So the hydrogen bonds that are holding that DNA double helix together are broken in the presence of the DNA polymerase. And that enables each strand to be functioning as a template. So the enzymes move in opposite directions as the replication fork opens. And the leading strand is a continuous strand. So that's replicated in the 5' to 3' direction. And the lagging strand is also replicated in the same direction, but in kind of fits and spurts, in that it's being made in these short fragments of nucleotides called Okazaki fragments. And those are kind of pieced together. So in that way, even though the terminus is not exactly at the origin, you're getting a complete copy. So the lagging strand is really going in the opposite direction of the leading strand, but because it's still polymerizing in the same direction, it has to kind of start polymerizing, and then it stops, and then it goes back again and polymerizes another fragment. So as the fork opens, it polymerizes these short fragments. And that happens in two directions, where you're having the leading and the lagging strand being polymerized, so that in the end you've got 4 strands of DNA, 2 complete copies of the genome by the time the enzymes get to the terminus. In the end, you have the parent strand here in black and the newly synthesized strand in purple. And there are other enzymes like ligases that fill in all of these gaps that you see between the Okazaki fragments to make a complete copy. And then the way that the cell can tell between the parent and the newly synthesized strand is important, because any mistakes that are made, it has to be able to tell which one was the original template strand and which one being the correct sequence, and then which one is the strand with the mistake. And the way it can tell is that DNA is methylated, but the newly formed strand is not yet methylated. So after the completion of the synthesis of the newly formed strand, then methylation occurs, and the separation of the 2 complete genomes occurs, as well as a modification. So there has to be an unwinding. There's some tension that builds up in the twisting or the helical shape of the DNA, and that tension has to be released in order for the genes to be able to be expressed. And that's those negative supercoils, or that kind of unwinding, is accomplished by a different set of enzymes, gyrases, and there are 2 major gyrases, A and B, in bacteria, that cleave both strands of the DNA, which requires energy in the form of ATP, and then they catalyze the unwinding, and then religate the 2 strands. And why I'm mentioning this in particular is that these enzymes are target of a class of antibiotics called quinolones, which are sometimes used to treat infections, bacterial infections. And it's a selective target because these enzymes are present in bacteria but not present in our own eukaryotic cells. So it's a good target for selective toxicity of antibiotics. So I'm giving you this information because it's relevant to the mutations, the mutation rate, and during this process of replication, there is a proofreading function that enzymes carry out in order to ensure that there's only, in rare cases, there's a mistake made in copying the genome. So the microorganism can survive, because if there are too many changes, it would result in malfunction of the products of the genes, and so the microorganism wouldn't be able to survive. But also it means that because these enzymes are not perfect in every case, although they're rare, but it means that changes can result in these rare cases that could lead to a different strain within the same species. So during DNA replication, there's about one error in every 1,000, but those errors are often corrected by these DNA repair enzymes that are able to detect mismatched bases, so they're not properly hydrogen bonding, they're not lined up properly within the DNA double helix to be able to hydrogen bond, and so they're recognized and they're repaired. And DNA polymerase 1 is one of those enzymes that detects those mistakes. And with the help of these repair enzymes, they are corrected. All right, I'm just going to touch briefly upon biotechnology before we finish today. There's tons of uses of bacteria and bacterial products that can be used to solve a problem, and that's really what biotechnology does. But I just want to, so even though these bacteria or their products can be really key tools that we use for biotechnology and for molecular biology, whether it's directly directly to solve a problem, or just part of the analysis that we do regularly on a regular basis within the research lab, or within the diagnostic microbiology lab. These tools, I'm just going to touch upon a few of these key tools. So by understanding the ribosomal RNA sequence, so using molecular techniques in order to determine a conserved sequence, a small part of the 5S ribosomal RNA. We can, sorry, not the 5S. The 5S is too small. It's the 16S ribosomal RNA, which has a sequence within it that's long enough, but very conserved, i.e. it doesn't change much from one genus of bacterium to the next. It really helps us to classify differences. In fact, before this technique was developed of sequencing the ribosomal RNA from the 16S part subunit of the ribosomal RNA in bacteria, we actually had completely different species that were lumped into one group, because we didn't understand that on the molecular level they were different than what we saw in the phenotypic level. And so this is really key for classifying and understanding differences between these types of or species of bacteria, as well as for finding smaller differences within a same species. So here on the left you see the phylogenetic tree, and there are isolates of the species of bacteria called Haemophilus influenza, which causes a kind of respiratory infection similar to the flu virus. Um, and you can see that there are lots of different strains with, or what are also called isolates within those that collection of species, Haemophilus influenza. Another way that we can identify bacteria, whether it's pathogenic or not, is by understanding its DNA sequence. So we can sequence the entire genome, but that takes quite a long time and a lot of money and effort. But by looking at regions within the genome, using probes here, DNA probes represented here as the purple arrow, that have been tagged with something that is detectable, either a fluorescing molecule or a radioactive molecule, we can see if there's a good match between that probe and the target DNA. And if there is, then we know it's complementary. Those complementary base pairs are allowing for that really good hydrogen bonding that keeps those two strands together, and we can amplify it by methods like polymerase chain reaction, and actually see that it's conserved, and we can do identification of microbes that are hard to grow in the lab. For example, some viruses are diagnosed by PCR reaction. And the enzymes that are used for the PCR process are actually from a very extremophile type of bacteria that can grow at very high temperatures. And so that enzyme has been purified from those special type of bacteria so that it can be used in the cycles of low temperature to high temperature without being destroyed in the process during PCR. So there are many, many examples besides just these two that are examples of how bacteria are used in biotechnology, and how molecular techniques such as these are used in diagnostics. And finally, I'm just ending with a few suggested practice problems from the objectives review and the review questions.